And I'll turn this over to Dr. Anna Wald, who's the head of the Division of Allergy and Infectious Diseases, to introduce the Kirby lecture and the Kirby lecture for 2024. We're a few days late, but that's good. It's a 2024 lecture, but it's we're having it in the early part of 2025. So Anna, I'll turn it over to you.
Thank you, Conrad. So today's lecture is named in the honor of Dr. Bill Kirby. Bill Kirby was one of the original members of the UW Department of Medicine and the first infectious disease specialist in Pacific Northwest. His research focused on antibiotics, and he developed the standardized procedures for testing antibiotic susceptibility that have been adopted by the FDA and are still widely used. He was recognized as a master teacher and a clinician. Can I have the next slide, please? And in honor of Dr. Kirby, his family endowed a visiting professorship in infectious diseases. The criteria for selecting Kirby visiting professorship are qualities that are modeled after Dr. Kirby. So intellectual curiosity, scientific excellence, superb clinical and teaching skills, and national leadership. And as you can see on the next slide here, we have had quite a presentation from most of the renowned infectious disease leaders in the field over the last 30 or so years. So it gives me great pleasure today to welcome Dr. Hana Mohammed Sahly, who will be our speaker. She visited us a couple of weeks ago and gave a wonderful presentation and met with the faculty and the trainees. Dr. Sahly is professor in the Department of Molecular Virology and Microbiology and Medicine, and Kyle and Josephine Morrow Chair, Department of Molecular Virology and Microbiology at Baylor College of Medicine. She directs the Baylor College Vaccine and Treatment Evaluation Unit, part of the NIAID network to evaluate novel vaccine and therapeutics for infectious diseases. She was the national co-chair for the Moderna COVID-19 study, so the first mRNA vaccine that we have, and participated in multiple other investigational vaccines and observational studies for SARS-CoV-2. She's also a lead vaccinologist in other respiratory infections, such as influenza and RSV. Dr. Sahly chairs the Food and Drug Administration Vaccine and Related Biological Advisory Committee, and she's the liaison of the FDA Vaccine and Related Biological Advisory Committee to the National Vaccine Advisory Committee. She has won multiple awards, including several for research symposia, is a fellow of the IDSA, and was recognized for her work on DEI at the National Diversity and Leadership Conference. And Dr. Sahly is the deputy editor of the New England Journal of Medicine, Journal Watch Infectious Diseases. So welcome, Dr. Sahly. You can please start sharing your slides now.
Well, thank you, Anna, for the kind introduction, and it's a great honor to be the Kirby lecturer for 2024.
And could I add that for the folks who are attending this lecture, please insert your questions if you have questions into the chat, and then Dr. David Rink, one of the chief residents, will pose those questions at the end of the presentation. Thank you.
Are we in the correct mode now? Are we in the correct mode?
Yes, yes, it looks great.
Very good. All right, so this afternoon we will be discussing, I'll be discussing with you Influenza A H5N1 outbreak, H5N1 of cattle, birds, and men. I have no conflicts of interest to disclose, and today I will be going over the virologic characteristics of flu H5N1 virus, provide an update on the epidemiology of the virus and the clinical characteristics of human infections as we know them so far, with an overview of the vaccine development successes and challenges. So the earliest description of what seems to be a highly pathogenic avian influenza took place in 1878, where researchers described a disease that resembles what we know today as avian influenza. However, the first positive identification of flu A H5N1 in poultry as HPAI occurred in Scotland, 1959. Here, I would like to emphasize that the pathogenic in HPAI refers to a phenotype in chicken. The virus is deadly in chicken, and that phenotype is usually detected by giving the virus intravenously to chicken and looking for the outcome of disease severity and mortality. At a molecular level, it has to do with the hemagglutinin having basic substitution that allow for more rapid cleavage and thus infectivity in the chicken. And so far, of all the 18 H subtypes, only the H5 and H7 viruses have evolved to be highly pathogenic avian influenza. Between 1959 and 1996, it was no big deal. There were 14, 15 small sporadic epizootic recorded. You can argue maybe we didn't search hard enough, but that's the record out there. But since 1996, the epizootics seem to have become more frequent, and they involved more avian species. And we came to learn since then that the migratory wild birds are the vehicle of dissemination of the flu A flu AH5 and the H7 as well. Precisely because many of them have no disease. It's either subclinical or asymptomatic, so they can transmit the virus across continent during their migration. And since 1996 and up until the early 2000s, the disease in domestic bird, as represented here in the bottom panel, seems to have centered mostly in Asia. The red columns represent Asia. And slowly beginning to have some European and African, mostly in Egypt, outbreaks in domestic birds in the mid-2000s onward. And when it comes to the wild bird in the top panel, we see that there was some spillage into Europe in the early 2000s, but then mostly small numbers of outbreaks in wild birds and mostly still in Asia. In 2014, that has changed, whereby most of the wild birds detection and dissemination of the virus shifted to Europe, probably via Egypt, but shifted to Europe as depicted here in the wild birds outbreaks in green. And fewer of them were detected in Asia. And at the same time, this was largely driven by the emergence of the clade 2344, which is a constellation of H5NX. It was H5N1, N2, N6, N8, multiple H5NX subtypes. And they started causing more outbreaks in wild birds and domestic birds. But in late 2021, the HPAI H5N1 that is circulating now, the 2344B clade, replaced most of these H5NXs. And the spread became more accelerated since 2021, involving all 5 continents, with incidental infections in mink farms, certain mammals, and carnivores. And the evolution, for the first time, the evolutionary center of the H5 shifted from Asia to Europe. Also, since then, since late 2021, the spillover into mammals accelerated. And it was of 3 different varieties. One was the individual dead-end spillover into large cats, foxes, certain individual marine mammal. There was a big outbreak of cat food in Eastern Europe. But all of these were spillover individual events with no transmission among mammals. And there was the mink farm outbreak in Spain. Here, it's unclear how the transmission occurred on the farm. But then in 2022, there were events that could only be explained by mammal-to-mammal transmission. And these include the large die-offs of the marine mammals in South America and the current dairy cattle outbreak in the United States. So what happened in South America? It's a very interesting figure. You can see a wave coming from the west coast of South America in time and in species, going down the west coast and up the east coast, whereby infection in birds detected in 2022 and late 2022 resulted in spillover to mammals, but then there were large mammal die-offs in between that occurred in a spatiotemporal wave in South America. Genotypic analysis of this particular outbreak did show that there is adaptation in one of the genes, the polymerase basic, that is associated with replication in mammals. And then the United States outbreak. In January 2024, farmers in Texas noticed that their dairy herd are producing less milk and that there were dead cats on the farm. In March 2024, clade 2344B, the same one that caused the large mammalian die-offs in South America, was confirmed in the dairy cattle. And the genetic analysis revealed that a single genotype is causing all, the outbreak in the dairy cattle, which is the genotype B313, as a single introductory event from migratory birds. The hypothesis is that two genotypes, the B39 and the minor 5-1, did reassort in an unknown host, and then resulted in this current genotype, which was first detected in Wyoming, Colorado, and California in birds prior to its detection in the cattle. But once it got to the cattle, it got amplified with cattle-to-cattle transmission. And this is very interesting because cows are not usual influenza A virus hosts. These affected cows have poor oral intake, respiratory distress, and decreased milk production. But there is cow-to-cow transmission. And this is evidenced by when healthy cows become infected when in contact with sick cows that were transported from other states. And it's important to know here that not all cows are equally susceptible. It's the lactating cows that are susceptible. The virus, as it is right now, has high tropism for the alveoli of the mammary gland. When a regular calf is infected, there is replication in the upper airway. They might have minor symptoms, but they won't transmit, and the disease is self-limiting. But when a lactating cow is infected, the virus will replicate to high numbers in the mammary gland tissues and gives the phenotype that we currently are describing. The cow-to-cow transmission seems to be highly facilitated by milking equipment. The virus does survive on the milking equipment. It was detected, infectious virus. And when milking multiple cows, they don't clean in between. It's the milking equipment seems to have potentiated this outbreak, and of course the interstate transport, resulting in the federal order to test the dairy cattle prior to transport across states in May 2024. So how did humans come to be infected? Again, as far as we know, this particular genotype, B313, was first detected in wild bird and other animals. It spilled over into cattle. Then the current agricultural practices of transporting, the cattle across lines, of using same milking equipment, resulted in the cattle-to-cattle transmission, with spillover into other animals and spillover into humans. The humans could be infected directly from the lactating cow, or when the cow transmits it to an animal, such as a cat, the human can become infected from that secondary animal. So all of this talking about the lactating cow, how safe is our milk supply for consumption? Sampling the retail pasteurized milk from 17 states revealed that 20% of the samples, random samples, are positive for the H5N1 nucleic acid RNA. However, no infectious virus was retrieved from these retail pasteurized milk. So the widely used pasteurization methods do work. Raw milk is a different story. Raw milk does have infectious virus, and when stored at refrigerator temperature, even up to weeks, the viral load will decrease, but it does not eliminate, storage at low temperature does not eliminate the infectious virus. It seems that producing yogurt and cheeses from raw milk, because there's a lot of boiling involved, etc., it does inactivate the virus, but that's under experimental conditions. So it's still not recommended to consume raw milk in dairy. Where do we stand right now? There are 913 confirmed infections in dairy cattle in 16 states. There are 1,346 reported outbreaks in aquatic birds, commercial poultry, or hobby and backyard flocks. 582 counties are affected in all 50 states. What about infections in humans? We're going to break this into two periods. 1997, when the first human case was detected, till 2022, when this clade emerged, and then 2022 and after. Between 1997 and 2022, there were close to 897 reported and confirmed human infections with flu A H5N1. Most of the cases were severe and resulted in mortality of 56%. We can see that they had a relatively large geographic spread, initially centered in Asia, with China being the location of the first detection. Many cases in Vietnam, depicted in the dark green here. The light green next to it is Indonesia. And then many cases in Egypt in the 2010 onward. The current outbreak after 2022, there was one case in Colorado in 2022 and everything else in 2024. The current US outbreak has, as of the 30th of December, 66 confirmed human infections. 40 of these 66 were exposed to poultry, 24 were exposed to birds, and 2 had unknown exposure. There's no documented human-to-human transmission yet. The disease severity, in contradistinction to the previous reports, was mild. There was one severe case in Louisiana, and all other 65 were mild cases. There's also evidence of subclinical disease in the dairy workers in a survey, in a sero survey in Colorado among dairy workers. 7% was positive with evidence of recent infection. Only 4 of these 8 people recalled an illness, and it was mild and self-limiting. However, worldwide, and in addition to the Louisiana case, there are 5 confirmed severe cases since 2022, 1 in Canada, 2 in China, 1 in Ecuador, and 1 in Chile. So why the difference in this disease severity? We did say 56% mortality between 97 and 2022, while here it seems to be mild for the most part. There probably is some ascertainment bias here. Many of the cases, those 66 cases, were as a result of investigation and looking for these cases, while the other ones sort of percolated because they were severe. So there's a lot we don't know yet. Did the viruses change its virulence as a result of being transmitted and evolving all these years? That is unknown. And the interesting other feature here is the predominance of conjunctivitis in the many of the cases reported so far in the US. Here we see that only 7%, or 3 out of 46 cases, or 45 cases, had no conjunctival symptoms. A third had conjunctival symptoms only. 31% has conjunctival symptoms with a respiratory symptom, and 29% had conjunctivitis with non-respiratory symptoms. The median number of days with symptoms ranged from 2 to 5, and almost all the cases in the US, 87% of them, were treated with oseltamivir at the range of 1 to 2.5 days post-symptom onset. So why did this H5N1 not become a pandemic yet? It's been in circulation since '59. It's evolving. It's spreading across continents. Well, it has to do, as far as we know, with certain viral characteristics that did not occur yet. And these have to do with having evolutions in certain steps leading to transmissions, to higher transmission in humans at an R0 of greater than 1. 2 of these features have to do with the hemagglutinin, which is the attachment and viral entry protein depicted here in purple on the schematic. It has to do with that particular protein receptor binding tropism and the stability of this protein, the HA protein, in acidic pH. Then there is the polymerase changes resulting in the utilization of human cellular proteins for replications efficiently, and changes in the nucleoprotein resulting in evasion of human innate immunity. As far as we know, all these 4 elements have occurred sporadically, but never consistently and never transmission so far. So let's explain what they are briefly. The receptor tropism. Influenza viruses attach to their host cells using the HA by attaching to silosaccharides on the cell surface of the host. And human influenza viruses bind efficiently to those sialic acids with an alpha-2,6 configuration. The avian influenza viruses prefer the alpha-2,3 configuration. So far, all the viruses in circulation have preferred the alpha-2,3 configuration. And it's not like humans don't have the alpha-2,3 configuration. It's just a difference in abundance. The upper airway is rich in alpha-2,6. The lungs and the lower airways are rich in alpha-2,3. The eye is rich in alpha-2,3, and which explains a lot of the conjunctival symptoms we've seen so far with the current outbreak. The a single mutation introduced in vitro at position 226 can switch this binding specificity from the avian, which is the 2,3, to the human, 2,6. But it did not happen yet in the wild. A recent report that just released in the from the CDC showing deep sequencing of the H5N1 isolate from the severe case in Louisiana did show that there were low frequency mutations at positions 134, 182, and 186 that improve the binding to the alpha-2,6. And these same changes were seen in the severe case in Canada. But no virus in the wild and no domestic birds in Louisiana or elsewhere seem to carry these particular mutations, and certainly not carry them forward. So the hypothesis is that these low-level mutations seem to have developed in the host themselves, again, at low frequency. The other important change that needs to happen for efficiency for the virus to become a pandemic is changes in the polymerase proteins, the basic and the acidic, allowing the virus to utilize the machinery of mammalian host effectively for replication. Some of these mutations have occurred already at position 627 and 701, and these were detected in the large mammalian die-offs, but these changes are not consistent. However, as opposed to the HA, it seems that the evolutionary barrier to acquiring these mutations is low. And the third important change that needs to happen in the virus for it to become, or at least has the potential to become, a pandemic, is a stability, increasing the stability of the HA at lower pH. Because the HA changes conformation at low pH when it is in the endocytosed vesicle. However, this same change can occur when the virus is carried in droplets and transmitted. And if this conformation happens prematurely, it will result in loss of infectivity. So at the moment, the H5N1 HA is not stable enough to transmit in the upper airways. The genetic changes that can alter this phenotype are a little harder to predict, and they seem to be spread out throughout the HA length. So seems like a lot needs to happen for this virus to become a pandemic. Between the HA, the polymerase, the host immune system evasion, and the pH stability. And they seem to have been to be under evolutionary constraints because it didn't happen despite all the spread across continents. However, flu has its own shortcut to bypass these constraints, and it's the reassortment pathway. And the more flu A infects hosts that are usual hosts for flu A, the higher the likelihood of reassortment. Examples of such hosts are the horses, dogs, pigs, which seem to have triggered a couple of pandemics, reassortments in pigs, humans, and of course, birds. So where are we with the risk of transmission or transition to a pandemic? The CDC and the WHO tell us that the risk is low. The CDC has a tool called IRAT. The WHO has a tool called TIPRA, but they all have three components. One that relates to the virus, and the virus doesn't seem to be there yet. One has to do with the immunity of the human population, and we are very susceptible. So that would be increasing the impact of a pandemic. And the ecology, which is how how spread this particular virus is across continents, across species, etc., or in an appropriate species. And that seems to be the concerning part. The more species are infected, the more mammalian to mammalian transmission is being confirmed. That seems to be the concerning part, but not yet. A word about the treatment and prevention of avian influenza. The good news is that all sequenced viral isolates from the current outbreak are expected to have full in vivo susceptibility to oseltamivir. 3 human cases have harbored some neuraminidase mutation. As you know, the oseltamivir is a neuraminidase inhibitor. 3 human cases have a particular mutation that results in a 6-fold increase in the IC50 for the oseltamivir, but usually it's not considered to be clinically meaningful unless it's more than 10. So oseltamivir is expected to retain full clinical activity even with this mutation. Baloxavir, there was 1 human isolate that carried a mutation, which is the I38M. This one results in H3N2 isolates and a 10-fold increase in the IC50. So maybe it has just a borderline effect on its susceptibility. And so what do we know about treatment and efficacy or effectiveness of treatment with neuraminidase inhibitors in Influenza A H5N1 infections in humans? No clinical trials have been performed to answer this particular question. All we have is retrospective data, and many of it in different, in other countries. There was an Indonesian outbreak between 2005 and 2007. 127 patients were affected. 81% of them died. In a univariate analysis, increasing the time to treatment was associated with increasing mortality. And in a multivariate analysis, being part of a cluster was associated with decreased mortality, probably because it's related to time to treatment. Of course, they don't have the numbers to answer the questions, but from this figure, we can see that the best outcome, and it's based on 1 patient, was in the one who started treatment less than 2 days after symptom onset. But there is a gradation. The red is 2 to 4 days. The green is 5 to 6 days. The more than 6 days is the deep blue. So the longer, at least the trend is in the right direction. But given the rarity of the disease, this is all we have. In a study that also depended, that is retrospective and much like the previous one, but here, instead of data from 1 country, it relied on the global patient registry because it's a disease that's reportable to the WHO. If we focus on the 2nd column versus the 3rd column, getting oseltamivir at any time resulted in a 53% survival, while no antiviral treatment resulted in 24% survival. And if we look at this, also the 2nd column, we see that the longer it took to start oseltamivir, the lower the survival. So we take these data with a grain of salt, but it seems that all we can say is the sooner we start oseltamivir, the better the outcome, understanding the limitations of these studies due mostly to the rarity of the disease. So what is the current guidance? Antiviral treatment is recommended as soon as possible for outpatient or inpatient individuals who are suspected, probable, or confirmed cases of influenza A H5N1, The treatment is the same one as for seasonal flu, which is oseltamivir 75 milligram twice a day for 5 days or 10 days if the patient is critically ill. If the patient is hospitalized or if they are immune compromised, it's highly recommended that, or it says consider, but strongly consider combination therapy with oseltamivir and baloxavir to decrease the risk of emergence of resistance and to lower the duration of viral shedding. Even in the Canadian case, whom we know now survived and recovered, they added amantadine, so they used 3 drugs. What about post-exposure prophylaxis? If someone has been within 6 feet of birds or other animals with confirmed flu A, H5 or H7, but what's circulating now is H5, monitoring symptoms needs to happen, and that is respiratory, systemic, and GI symptoms, and adding and considering prophylaxis if the exposure occurred less than 48 hours prior, especially if the patient did not have PPE on and was exposed, say, in a closed door barn or in close proximity to an infected animal. Here, the recommendation is not to give the prophylactic dose of oseltamivir, but the full treatment dose to decrease the chances of emergence of resistance. What about vaccines? Well, as we discussed before, we've been preparing for the worst when it comes to H5N1 since the early 2000s, and there are a few things we came to learn. First, we tried tackling the H5N1 vaccination as we do seasonal vaccinations, which is with whole inactivated virus. However, the avian the H5 and H7, but talking about H5 now, is poorly immunogenic in humans. For example, every year we get 15 microgram per flu antigen in our seasonal flu, but it takes 2 doses of 90 micrograms of the HA antigen to have 54%, just above 50, of individuals having a titer of HAI that is greater than 40. A titer of greater than 40 is what we consider to be seroprotection or putatively protective from influenza. So it needs high doses of HA and it needs 2 injections. And in a pandemic, that is hard. So for influenza A, H5N1, achieving optimal immunogenicity requires the use of adjuvants. Many studies have demonstrated that, and one of them is the one in this slide. Here in a study, we randomized 225 individuals to get 2 doses of inactivated vaccines, the Indonesia clade, at 3 doses, 3.75, 7.15, and 15. With or without the ASO3 adjuvant. Without an adjuvant, excuse me. Without an adjuvant, seroprotection was no more than 4 to 8%. With the addition of an adjuvant, 86 to 100% became seroprotected or achieved seroprotective titers. And it's depicted here in the figure with the dramatic rise in HAI titers. What we also can see is that when you use ASO3, very little antigen is needed, and there isn't much of a difference between 15 or 7.5 or 3.75. So you can have a lot of dose sparing that way, which can come handy in a pandemic. Well, what about the currently circulating H5N1. Prior to the 2344B, there was a 2344C that was circulating in, I want to say 2017, 2018. Here we did a study using that particular strain, and this strain is very closely related to the current outbreak. The same result was seen, meaning if we give 15 microgram of the H5 without an adjuvant, only 25%, here on the third row, 25% of participants will have seroprotective titers. But if we add ASO3 adjuvant, the seroprotection rate goes up to 85%. And if we use MF59, it goes to 44%. Increasing the antigen dose to 15 with ASO3 doesn't improve the titers. You're still, well, it's still 85% protection, and it goes up to 65 in MF59. So all in all, an adjuvant is needed, and it seems that ASO3 result in more potent immunogenicity than MF59, and that has been demonstrated in numerous other studies with H5 and H7. And if we are to use an adjuvant, here we use ASO3 or MF59. Again, and again, the vaccine used has the 2344C. There's a high cross protection to the 2344B Astrakhan. 2344B Astrakhan is the one that is closest to what is circulating now in dairy, in humans, and in poultry. So we did develop and tested a vaccine that seems to cross-protect. Having said that, now the Astrakhan itself as an antigen is being used to develop a vaccine, and studies did start with that. The other benefit of using an adjuvant, in addition to improving the immunogenicity against the vaccine strain, is that it improves the breadth. Here, 22 adults who got 2 doses of H5N1, the Indonesia clade, got it with or without ASO3. In the unadjuvanted, we see that the titers were very low against the Indonesia, which was in the vaccine, or Astrakhan, which is similar to the circulating one. But when an adjuvant is used, and here it's ASO3, the titers are boosted against Indonesia, but also against the current strain. We attempted also other modalities of improving the immunogenicity of H5N1. Also in the mid-2000s, there were many studies that have hypothesized that giving the vaccine intradermally will improve the immunogenicity because the skin is rich in antigen-presenting cells. So we tested this hypothesis, and we gave 227 adults 2 doses of the Vietnam vaccine, either intradermally or intramuscularly. What we found is that and this study was powered to detect a difference. The difference in the titers were very minimal, 18 versus 25, the seroprotection 35 versus 42. So giving the vaccine intradermally didn't seem to improve the immunogenicity. Another way to improve the immunogenicity also had to do with giving topical adjuvants, and there was a study in the 2015-2016 time where the seasonal influenza was given intradermally with a TLR7 agonist. which biases for Th1 responses. And there was a dramatic increase in the response to seasonal influenza when given in that way. So we tested what if we try this with H5N1. And so we randomized 50 participants to 2 doses of the imicomod cream with flu vaccine or control cream with flu H5 vaccine. And what we found is that there was really no statistical difference in the titers, 44% in the control arms versus 24% in the intervention arm had seroprotective titers. So if anything, numerically, it's in favor of the control cream, but safe to say there was no boosting of the immune response. Another important thing to think about in terms of pandemic preparedness is investment in population immunity. The identity of the flu subtype or strain resulting in a pandemic is not known in advance. One proposed approach is to vaccinate against the avian strain that's in circulation. And then whenever an outbreak or a pandemic erupts, then you can boost with a single dose of the matching strain. Because such an approach will maximize resource utilization and address time constraints during a pandemic. Of course, this is borne by a lot of the high mortality that was earlier observed with H5N1 and H7N9 in the range of 50 to 60%, as opposed to, say, the 5% that we saw with the COVID, which would be devastating if we need to give 2 doses even. And there is evidence to that. So participants who got 1 or 2 doses of the Vietnam clade at 90 microgram, we boosted them 22 months later with a different clade. And here we see, focusing on the Henri antigen, which is the one in the booster, examining the numbers with a titer of greater than 40, we see that boosting with an adjuvant, whether the patient got 1 dose or 2 doses, results in a 50% seroprotective titers against the current vaccine, but higher likelihood of response to the Vietnam, which is as a result of immune imprinting. But regardless, only giving 1 dose or 2 doses as a priming resulted in immunologic priming, although probably the immune responses were low to the original. And when you come and boost again, all you need is 1 dose. And we demonstrated that also with H7, the same phenomenon. Actually, here in 2008, a study of a flu H7N7 resulted in 0 to 5% seroprotection, 2 doses up to 90 micrograms. When we came 8 years later and we gave them a single dose of 45 microgram, we did note a rapid rise in hemagglutination titers and microneutralization titers that proves that immune priming did exist, even in individuals who had 0 responses in 2008. And with the current outbreak, more creative methods are being utilized to tackle the current pandemic. mRNA vaccines are under evaluations. Multiple other platforms are under evaluation. But here we want to turn your attention to a new way of potentially tackling drift. It remains a hypothesis, which is using a high-resolution antigenic map investigators designed a hemagglutinin with broad cross-reactivity potential. The ferret sera raised against a naturally occurring broad antigen, which is Iraq in panel A, versus a designed central antigen, which is CBA Anhui, CBA Vietnam, are depicted here. What we see in the Iraq one, which is a naturally occurring and the most central of the new H5s, it had 50 detectable titers to all the circulating H5N1s versus 82 detectable titers to the one that is designed antigen, or 78, which is the Vietnam. This hypothesis is about to be tested in humans, and hopefully we will have something to share maybe in a year about that. Another important piece of the puzzle that seems to be neglected in flu vaccines, whether the seasonal or the pandemic variety, is the neuraminidase. We knew about the importance of neuraminidase for decades. During the 2009 and 2011, a study by Couch et al. Demonstrated that NAI inhibiting antibodies correlate with infection, with influenza illness and influenza severity. In a randomized vaccine trial, whether testing live attenuated versus inactivated versus placebo, all vaccines were less likely to produce NAI, but as the titer of NAI antibody increased, the risk of influenza infection decreased. And in a controlled human infection model, this has been demonstrated as well. Current vaccines have small, variable, and non-standardized neuraminidase content, except for the recombinant flu, which has none. We have done a study where individuals were randomized to increasing doses of the inactivated vaccine with ASO3 or without. What we found is that, and that was against the H5N1, all participants had detectable N9 at baseline, and it increased with age. However, the N9 NAI geometric mean titers increased in a vaccine dose-dependent fashion and with the inclusion of an adjuvant, whether in the young or the old, as seen on the right side here. So what about pre-existing immunity? I couldn't find much here, but there's an interesting study in ferrets where ferrets who were infected previously with the pandemic H1N1 versus naive ferrets were both challenged with the bovine H5N1 that's circulating. The ones that have been infected with the H1N1 before, depicted here in the red, had 100% survival, had less weight changes, and the cumulative symptom score was very low, which is very different than the immunologically naive ferret. It's a small sample size study. It is suggestive of a protective role of previous infections against viral replication, severity, and mortality of the disease. Mechanism, is it the anti-neuraminidase? Is it the anti-HA stock? Is unknown. And there's no evidence in humans yet or an understanding of the role of previous infections or the imprinting patterns. So in summary, H5N1 viruses continue to evolve and are infecting more mammalian species. Mammalian adaptations are occasionally occurring, but not consistently. The risk to human populations remains low by CDC and WHO criteria. Inactivated or subunit vaccines are poorly immunogenic, but the use of oil and water adjuvants improves the immunogenicity of these vaccines. 2 doses are needed to achieve seroprotection in a majority of people, should we invest in population immunity? A lot of pros and cons to this idea. The neuraminidase as an antigen continues to be neglected as part of the influenza pandemic readiness. And novel platforms and novel antigenic determinants are being tested, and this is a nascent field. Probably we'll have more to say about this in a year. And I'm happy to take questions. I don't know what I did with time, but...
Thanks so much for your talk, Dr. El Sahly. We are actually right at the end of time, but so I do want to be respectful of everyone who's attended in their time, but I will ask you one question for those who are able to hang around for it. One of the questions from the audience is that overall the sense from the data you presented so far is that the risk of bird or avian flu pandemic arising from the current virus is relatively low, and there seems to be some evidence of effective treatment options already. However, significant funds, over $300 million, were just allocated to prepare for this possibility of a pandemic. And wondering if you could clarify maybe some of the rationale behind that and what work that may be used for.
So this falls under the big umbrella of pandemic preparedness. Yes, as it stands right now, it is not likely, this current iteration of this virus is not causing a pandemic or likely to cause a pandemic, given the viral characteristics. But reassortment can happen fast. And if it were to happen, then one needs to be ready for that contingency. That's my take. And of all the ways our society can be spending its resources, human health is to me at least, a priority. And as we came to, I never knew this is how we milked cows and how we we need to invest in more ecologically friendly practices of agriculture, and that's probably a bigger problem to tackle.
Certainly many layers to this. Yeah. Well, thank you so much for taking us through that. Again, I appreciate your work, and thank you for being our Kirby speaker this year. Thanks everyone for attending Grand Rounds today, and we'll see you again in a couple of weeks.
I have my email. If you have the questions in the chat, I'm happy to answer them in an email and send them back to you or provide references.
Fantastic. Thanks so much.